course, the freshman semester, I guess. I mean, there's some smaller ones down the road, but this is a big 5-block, 10 lecture per block course. Out of curiosity, I'm just wondering, how many of you guys have had a microbiology course at some point in time? Ooh, a lot of you, like the vast majority of you. Okay, so this will be like a cakewalk. Right? Awesome. Okay, well, we'll go over how this course is set up, and I will try and kind of ping you guys a little bit. I'd like to get you to know you with the amount of time that we're going to be spending together. That would be a good thing. Starting with, at some point, maybe not right now, but you guys, if you would, take a piece of paper, fold it into a little triangle, and if you would just write your name on it or how you prefer to be called on that so we can see it. Oftentimes you guys are just wearing t-shirts anyway, so it's harder for us to get to know you. I know a couple of you a little bit. Sorry, you're John now, by the way. So I prefer calling you guys by your first names, or sort of manage to get to know who you are. And you don't necessarily have to do that. But go ahead, if you want. Let's see. I guess as you're doing that, I'll tell you a little bit about myself, which again, like I wouldn't normally bother with, but we will spend a lot of time together, and it'll help you know like what to ask me, what not to ask me. A lot of people are like, oh, how did you study for this when you were going through IPAP? Didn't go through IPAP. Not a PA. Microbiologist, someone else. So I do not know. I went through these places to get my education. Entirely different format. For my bachelor's, got my bachelor's degree in biochemistry. Can you buy anyone in Champaign or Van Natives? Yes? Sweet. Okay, cool. Yeah. Interesting, so I'm a native of Illinois myself, but I call Madison home. Anyone from Madison, Wisconsin?
I'm from Gurnee, Illinois, Chicago.
Okay, cool. Yeah. All right. Interesting. And I know when I talked to you guys last week during orientation, Kelly asked how many like San Antonio natives there were. A couple of you guys. So as the course progresses, you'll get a sense of my feelings about Texas as a general state for infectious disease. It's one of the most interesting states in the country. So we'll talk about that quite a bit. Okay, so yeah, home of record, I claim Madison. I've been in for a long, long time, since I was 17 years old, like right after I graduated high school. Enlisted for a long time at the Chemical Corps, and that's probably where I got interested in biology, I guess, NBC, nuclear biological chemical. So, did 4 years active duty in the Chemical Corps, and then went into the reserves so I could go to college and get my degree. And then I decided, after a long time as an NCO, I decided to drop a packet and commission, so I became an engineer for a couple years. Very convoluted career path, don't recommend it. Any engineers here? Yeah? Yeah? Spent some time in Leonard Wood?
Oh yeah, lots of them.
Awesome. Yeah, so did that for a while, and then finally, by that time I had, this is a bare minimum requirement to get into the AMED as a microbiologist, you have to have a master's degree. And since then the standards have increased, so you have to have a PhD to get in now. So I wouldn't have been eligible at the time. But dropped the packet once I got my master's in clinical micro, got picked up for microbiology. And I tell you what, we are a strange breed of cat, Army microbiologists. You guys ever worked with microbiologists before? A couple? Yeah? One? which one was it?
I don't know which one.
Okay. Are you a kilo?
No, I'm
Okay. Okay. Are there any lab techs here?
My degree is in medical technology, but I didn't really work at it. I joined Yagi right after college.
Oh, okay. Okay, so you no like legit 68 kilos here. Okay. All right, that's cool. Fewer people for me to pick on through the course. But if if you, like, I would be interested in what you have to say, like if you've had extensive training in lab throughout this course. Went to CGSC, picked up another master's in military operational science, and then finally through LPED, I got my PhD at the University of Virginia. Anyone that spent time in Charlottesville? Yeah?
Yeah, I grew up there until like third, fourth grade.
Oh, okay, cool. Yeah, beautiful part of the country.
Yeah.
Yeah. Yep, and good school as well. So yeah, that's me in a nutshell. I don't have any medical training, so I've got to muddle through anatomy, got to muddle through pathology, and all these tangential topics that we talk about as we talk about these different microbiology concepts. But we'll make do. It'll be fine. In terms of where I habitate down here in Medco, I'm down on the first floor, and you can come find me from 1317. That's in the southeast corner of this building, kind of right next to the other 71 series instructors. So if you've been down to Major Johnson's office in that area. Office hours are a little squishy for me. I don't like being in this building more often than I have to be, as you can imagine. So I am usually here 5 days a week. However, this noon to 1300 thing, you've probably heard this from other instructors as well. Like, we're almost always here during that time, but we also have a lot of meetings that are packed into that lunchtime time. So when you guys are available, we try to be available, but there's just a lot of meetings. So if you want to come and talk to me about exams or material or whatever, shoot me a text if it's urgent. This is my cell number, or email me, army.mil, or message me on campus. All of those are fine, don't care. So multiple ways to get hold of me. Okay, and then getting into this material, which hopefully all of you guys were able to access. Okay, anyone not able to access those materials? Okay, super. I will try to upload PowerPoints and PDFs because I've heard people have issues with PowerPoints a lot of times, so you should be able to find most of those most of the time. For a little bit of additional atmospherics, again, 5-block course, it has traditionally in the last, 6 or 7 years, been split taught by a civilian, Mr. Murray. You may have heard this man, he's kind of legendary. He has been teaching here for 50 plus years. He just retired last semester, so he was teaching this block that we're going to start off with, block 1. And then the green suit, or whoever occupied, my slot, was teaching blocks 2 through 5. So I've been here for like a little over a year and a half, and I've had time to revamp a lot of blocks 2 through 5. But block 1, I'm trying very desperately, as hard as I can, to get those slides updated. And that's why there's been a lag in getting this deck as well as the second deck. Block 1 is only a 2-deck block. It's this slide deck, which is like 80 slides, and then a big immunology deck, which has been a beast. So, in full disclosure, it needed to get updated. Some of those figures look like they were 20 or 30 years old. So that's just taking a lot of time, and I appreciate your patience. We'll try to go light today and tomorrow, possibly finish this deck on Monday, and then be ready for the immunology deck on Tuesday. Okay. But this block, if you look at the schedule, is pretty well spaced out. So I think we'll have the luxurious flexibility. Okay, so a little bit of stuff that you've probably already figured out. This is our course homepage. Got all the block course material loaded on these hyperlinks here. And so far there's just that one deck in block 1. We'll go over the syllabus in more detail, but there it is in like a rough syllabus left here, or on top. And then the course textbook, the Patrick Murray Medical Microbiology textbook, 9th edition, which came out in 2021, right after COVID. So it has a little bit of information about COVID, but not a ton. Okay, this is a little bit more of a detailed syllabus of how this course is broken down. Block 1, as I said, there are 2 decks. The majority of the material is going to be in that second deck on immunology. Block 2 is similar to block 1. It's still a little bit of intro, concept heavy, but important stuff, like microbial genetics. You gotta know a thing or two about bacteria, and this is oriented at bacteria, about how bacteria replicate, and in order to have any understanding of how antibiotics that target those processes work. So it's important that we have some foundational knowledge here. Control of microbes, how to kill them with antibiotics, how to kill them with disinfectants, how to kill them, how to sterilize them. We're talking about medical equipment, stuff like that. Tools and techniques of microlab. So this is a very superficial treatment of that. You'll get a deeper layer next semester when you do Clean Lab with Major Guzman, or I guess his replacement since he's deploying. But there's a whole block of Clean Lab that will talk about those. But we will get you at least up to speed, so when we're talking about PCRs and immunoassays and things like that, you're not totally lost. That's what we're talking about. So a little bit of block 2 for that. And then finally, probably the most important part is host microbe relations, because that is what sets you up for block 3, which is bacteriology. So host microbe relationships is talking about virulence factors, the balance between being in a healthy state and a disease state. It's just a lot of really good, important stuff. And then from blocks 3 through 5, we are, if you observe here, we're talking about the actual microorganisms. That's all we're doing for the rest of the semester. We're trying to familiarize you guys with pathogenesis, identification, and the main core things about these different microorganisms. Some of them you will have heard about before. Everyone's heard about Staphylococcus before. Some of them are pretty exotic. You probably will never encounter again, but you still kind of have to know about them in order to go out and be a provider. So we'll talk about all of those. And then in block 4, we start off with like a mop-up of bacteria, a category called atypical bacteria, which include things like Mycobacterium tuberculosis and some other things. But then we transition into viruses, which is the bulk of block 4. So we do a lot of virology. Oh, and I forgot to mention, by the way, that's my background. I'm a virologist. So not a bacteriologist, not an immunologist, not a parasitologist, not a mycologist, not studying fungi. This is where I live. Teeny tiny little piece. Then we have like one day of talking about fungal diseases, fungal pathogens, and then block 5. So block 5 is, I guess, what everyone kind of freaks out about, but I think it's a fun block. Any of you guys ever done parasitology, like a parasitology course before? No? Okay. And I heard, so we finished off with like one lecture on ectoparasitology. I heard there is an entomologist in this cohort.
He is, but he's
But
doing
he's- Who's not here? Okay, well darn, I was gonna pick on him. So yeah, we have one lecture for ectoparasitology, which hopefully he will enjoy. So yeah, that is our syllabus in detail. I mentioned the textbook. This is an ebook. So if you go onto Stimson's website, you can get access to this through Army University. If you prefer reading text, great, do that. I was always that kind of student when I was going through school, where I just liked to read textbooks. But I understand you don't have time for that necessarily. It's a good resource if you want it. Also, the 10th edition just came out, 2026. Elsevier 10th edition of Micro. So that's what the cover looks like there. I really haven't started pulling material for the 10th edition. I don't think it's substantially different, but we do have an ebook subscription to that as well for Stimson, maybe through the Google in the future. In terms of course materials, the vast majority of the stuff is going to come from these PowerPoints. There used to be a whole bunch of handouts and auxiliary materials, but I feel the need to kind of streamline things and make them as easy as possible. So if all you do is read these slides to prepare for exams, you should be in good condition. You have access to the textbook if you want it. We do go through and review the lecture before exams, so that should be helpful. But by and large, this was done with the intention of making things easy and simple for then occasionally, like, you guys see my Canvas message I sent to the class? Okay, you saw that article I sent on chronic wasting disease? Okay, so like occasionally there might be stuff like that, random stuff like that, but mostly it's just the slides. What we're talking about throughout the course, again, we're talking about these different bacteria, what they do, and what their mechanisms of causing disease are. Pathogenesis, post-microbe relationships. In terms of your knowledge base, a common criticism of this course is the content is esoteric and why would I need to know this as a PA? Well, like, you kind of have to have somewhat of a foundation, right? You have to be able to have an understanding of the nature of these things that are causing infections in your patients, which you will see. You know this, right? You're gonna see a whole panoply of different diseases and infections in patients. So there are no easy answers. This material is difficult, and you have to kind of embrace that. I assume you all joined this program because you're willing to do something difficult. You don't enter medicine and you don't get into science unless you're ready for that, because these are complicated topics. And I tell you what, like, when I listen to a lot of infectious disease podcasts, and I listen to infectious disease doctors having these discussions about treating patients. A lot of the times they're talking about treating really complicated, interesting cases. Sometimes they're just talking about treating somebody who has like a staph infection, right? So you can't blow off something mundane like staph and think that it's easy. Those can be incredibly complicated as well. So I'm trying to get you guys to have a respect for the material and for the complexity of it without making it overly difficult. But just know that inherently microbiology is a hard subject, okay? You'll also need to not sound uneducated or imbecilic in front of your patients. I mean, don't embarrass yourself and have your patients know more about their infections than you do. So, patient education, being able to give them something of cognitive value when you're seeing them. You may end up doing something in the realm of forced health protection. Done a couple of assignments like that myself. So, you never know. You need to know something about microbiology for that reason. And also, this layered approach to this course. Let me go back to this slide. I mentioned that some of these dovetail with other courses that you'll have later on in phase 1, like this being tools and techniques fitting well with FinLab. All this material is meant to really dovetail with other courses. When you get to sophomore semester and you get into HE&T or radiology, for example, FinLab, some of those other courses, they're going to be talking about a lot of the same bugs that we're talking about now in this course. And they'll expand on them and they'll assume that you have that basic knowledge as well. So it should all build upon each other. Like skipping down to this bullet at the bottom here, not a medical treatment course. There has to be a delineation, and that's where I stop. I don't talk about treatment. We will talk about antibiotics a little bit to prep you for Pharm 1, your sophomore semester. But a lot of your treatment is going to come at your junior semester when you go into infectious diseases with Colonel Gutierrez, who is a PA. Okay. So that's another common misconception that we're just going to teach everything in this course. We talk about pathogens mostly here. And also, unfortunately, not a lab course. It would be super fun if we could get you into a lab and get you some hands-on time growing up bacteria and doing whatever that you'll never have to do as PAs, but not like that. I'm not going to do that. And then the redundancy, that should be a good thing. A lot of things in this deck are primers for subsequent decks that we'll cover. So redundancy is built into the course, and hopefully that's something that benefits you instead of bores you to tears. Okay, in terms of prep, I have a couple suggestions with this. We will go over TLOs and ELOs, but especially when we get into blocks 2 through 5, we won't go over them as much because they become a little bit repetitive. But still, when you ask yourself at the end of a block when an exam is coming up, what do I need to hit hard to study for this exam? Go back to those learning objectives and hit those hard. make sure that those concepts are what you're paying attention to. I've had students ask me like, do I have to memorize the date of, the first bubonic plague, blah, 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 the date range? No, I might mention something like that and, technically it would be testable, but the material that ends up on exams is designed to be the most bang for its buck, like major concepts. So I can't tell you what's on the exam. I don't like spoon feeding, drives me crazy. But if something's important, you'll know it, okay? Some classes, every class has its different dynamics. And I had a the previous -2, 24-2 was my class. So when they came through a year ago, they were a very shy group of people. And I think they had some group dynamics where they squashed students from, feeling comfortable asking questions. And that's a shame because the questions that you guys come up with are really, really beneficial for the entire group. They're things like clarifications that need to happen, broad questions, specific questions, things that I forget to mention. So if you feel comfortable bringing up those questions, it helps everybody, it's extremely beneficial. For study methods, I will say the same thing that everyone has been saying to you guys for the last 2 weeks, and that is make sure that you kind of stay on top of the material, especially after a lecture. I know that's tricky once you get into the rhythm of preparing for your Monday or Friday exams, but if you can spare just a little bit of time going over those slides we went over, maybe generating some questions of things that weren't clear to you, that should be super helpful. Okay. And then these are kind of Mr. Murray era pieces of advice, but if it helps you to draw things out, that does help the learning process. Transitions from passive to active learning, so that can be extremely helpful for some people as well. But, a lot of people ask me how they should study, and to that, I can give you some glib answer, but it depends on you, right? Whatever works best for you. So that you can use this semester to sort of figure out what your study techniques are, whether you're just watching YouTube videos, Ninja Nerd, whatever, group study, reading textbooks, whatever, they're all good. However you want to spend your time. But it is helpful to probe your own knowledge and make sure that you are able to look at a slide of information and answer the questions that you think would be important. Your ability to formulate questions for yourself. Okay, any questions on the way the course is set up? Yes, sir.
So, syllabus diagram, just the numbers associated with it, like I said, just kind of structure.
Great question. That's relative weight.
Right.
Yeah, so, yeah, 60-40 maybe, 70-30 split for block 1 type of thing. Just gives you a rough estimate, but not something you can, yeah.
We're not setting watches.
Say again?
We're not setting watches by it, so to speak.
Yeah, exactly, not setting watches by it. Yep, it's still fairly rough, just to give you a rough estimate of how much time we're going to spend on those different topics. Yeah. Okay, well, if there are no other questions, we'll hit the TLOs. This is very broad for this deck. What we're going to be doing is going over, and for the next deck, basic immunology in the next deck. Blocks 1 through 2, we'll cover those materials. We've already hit this, so very briefly. And then 3 through 5, which is about 60% of the course, that's when we'll cover the specific areas of microbiology. We will also be looking at the roles of these microorganisms, talk about their morphology a little bit and their culture characteristics when we're talking about bacteria, but we're not going to be teaching you how to be a lab tech or how to identify Staph aureus in the lab. Show you a couple plates and things like that, but most of the emphasis is on what is this pathogen and can you associate it with different diseases that it's capable of causing. We also hit the epidemiology of different diseases. If we're talking about chlamydia, we want to give you a sense of what percentage of the population is affected with that disease. Transmission, this is just basic education on these different diseases, how they cause disease, and a little bit about what kind of immune response is elicited from them. we hit most of this for the ELOs. Just with the general characteristics of these different pathogens that we're gonna be talking about. We'll try to hit most of these subcategories. Very, very light on lab diagnostics this block, but next block you can expect more tools and techniques. All right, so let's get into it. Couple slides just introducing you to microbial world that we live in, as evidenced on this pin, where you can see a perfectly clean looking steel pin might look like it has nothing growing on it or nothing capable of growing on it. But if you zoom in quite a bit, you can see that you have tons and tons of bacteria that are capable of adhering to stainless steel. So if you get a puncture wound from a pin that looks like it's sterile, rest assured it's not sterile. You're introducing bacteria into the skin that way. So that is why we flame sterilize at a minimum things that are steel that are penetrating into our bodies. In terms of the burden of microbes in the world, we have a number that I've never heard of before, 5 in a million. That's 5 with a string of 30 zeros behind it, in terms of the estimated numerical abundance of bacteria on the planet. Kind of like a mind-boggling figure. And that includes both in soil as well as out in the oceans, where if you ever get a chance to take a course on microbial ecology, super fascinating. But with that many bacteria, rest assured they're contributing heavily to the carbon shunt, the nitrogen shunt, recycling of nutrients in the world's oceans, for example. So major roles there. Life on Earth would cease to exist without them. Life wouldn't have begun in the first place without them. They're progenitors. Very, very important. And those are just the ecological bacteria. Getting into those that live in or on us, as most of you know, particularly our gut, our intestines are heavily populated with bacteria. There's this very common knowledge that you have about 10 times more bacteria in you than you do your own human cells. So numerical imbalance of prokaryotes to eukaryotes in yourself. And they are small relative to your cell size, but that is a lot of them, and that's just in the GI tract. You also have them on your skin, you have them in your mouth, you have them in your reproductive tracts, and some other areas in the body as well. The old iceberg analogy is useful for kind of understanding that we are only talking about a relatively small percentage of bacteria in this course. We're talking about the pathogenic ones mostly. A little bit of beneficial bacteria when we talk about gut health, but most of the bacteria in the world, just know, are not capable of harming us. They're avirulent, non-pathogenic, and there are some that are even beneficial. The ones that live in your gut, for example, they're the ones that are not just producing vitamins, synthesizing vitamins that we can't, but they have catabolic capabilities and digesting food that we can't do enzymatically. So they have that important role. Paradoxically, or I guess counterintuitively, they support the immune response. So this is what is teaching and training your own immune system what is friendly and what is not friendly. Without that, we would be completely at odds with the world. And then out in the world, they play an important role in just decomposition, of course, making sure everything's broken down in the environment, working with fungi, and we use them for food and beverage production. Beer, for example. Anybody know what brewer's yeast is? Scientific name for that? Yeast? You know? You know beer nerds? Saccharomyces cerevisiae. Okay, we'll know that. We will know that. It won't be a testable idea or a concept, but we'll know that by the end of the course. Okay, in terms of how we study microorganisms, this is pretty tedious stuff, but it's important to have at least a bit of a handle on how we categorize microorganisms. Starting with the definition of what taxonomy is. If you haven't heard of this term before, it means the classification schema for all biological entities, not just microbes, but all of life. So the science of biological classification. And we have a couple terms that are important to understand, be able to distinguish that fall under that. First is classification, how these organisms are arranged and grouped based off of mutual similarity or evolutionary relatedness. Talk about that in a little bit, but the formal classification system is kind of like how we understand different organisms. Nomenclature is another concept that I think is irritating to a lot of people, but it's important that we call things by their right name in the right convention. With all living cells, we have all species, we have a binomial nomenclature. Sorry, there's a P that was missing from two-part on your slide. But this dates back to the Swedish biologist Carl Linnaeus, if you've heard that name before, the 18th century biologist, who recognized that there was a need to have a systematic way of referring to organisms that would be the same here or there in Sweden as it is here in the US or in Japan or in South Africa or wherever in the world. So binomial classification schema has a genus, species arrangement or convention. Both the genus and the species are italicized, and very approachable example is Homo sapiens, us humans. So the genus is Homo, sapiens is species, the knowing modern man. Homo sapiens sapiens is a subspecies, so that's modern man, 150, 200,000 years old, something like that. And then the example of a bacterium that you've probably heard of before is E. coli, Escherichia coli. The genus is Escherichia, and there are other species, but the most common is coli. That's nomenclature for identification. That's what we're talking about when we're talking about how we figure out what these different bacteria or parasites or viruses are. So this is more of a practical aspect. We're talking about how we diagnose. Researchers need to be able to identify things, environmental biologists, all of them need to be able to figure out what is what. So that oftentimes is based on cellular characteristics. Going back to taxonomy, you've probably sometime in your careers as students seen this this pyramid before that arranges a hierarchy of different taxa. Taxa is the plural for different taxa in singular. So starting with very broadly domains at the top. Prokaryotes would fall into two different domains, archaea and bacteria. And eukaryotes have their own domain eukaryota. Anybody know what the difference is between prokaryotes and eukaryotes? Yeah, which one has nucleus? Eukaryotes, yes. The true kernel, the true nucleus, eukaryotic, and then prokaryotic before a nucleus was evolved. For microbiology, unfortunately, this is one of those areas that kind of, like a lot of things get lumped under microbiology. So we can't just be content to study bacteria. We also study in block 5, we'll talk about parasitology. So we'll go back into the eukaryotic domain. We'll talk about worms, which belong to kingdom Animalia. And we'll talk about protozoan parasites, which belong to kingdom Protista. And then kingdom fungi, that are fungal pathogens. For prokaryotes, we're not talking about archaea. They're clinically not significant for the most part. So we're talking about the domain of eubacteria, true bacteria. And these really don't have kingdoms anymore. They used to be in the kingdom Monera. That is obsolete now. So really it's more like it skips down to phylum level. Big phylums like Prophyla, Actinobacteria, Firmicutes, Bacteroides. For those of you who are health nuts and study, like the balance of bacteria in your gut, you might have heard of some of these before, but those are huge bacterial phyla. And then also, to kind of drive that point home, this is a taxonomic tree we're seeing with all 3 of these domains that I mentioned represented. Eukaryotes are just one branch of this tree, and then archaea and bacteria, as you can see, these are single-celled prokaryotes, but they are so different from each other. You would think that they would be similar, but they really aren't. Archaea are actually evolutionarily closer to us as eukaryotes than they are to bacteria. And a lot of that is due to the way they metabolize different energy sources, primarily. So here, this is where we are, one teeny tiny little branch. And the length of these branches roughly correlates to the length of time from divergence from these common ancestors. Wherever you find these nodes that meet, that's where the last common ancestor is. So this node, for example, at the very bottom, would be the last common ancestor between bacteria, archaea, and eukaryotes. So everything we're talking about in this course will fall under one of those three domains, with the exception of viruses. Viruses are not included in this schema because they're not living. So they have their own taxonomic schema, and we'll get into that more in block 4. There was an attempt to kind of smush them into that binomial classification schema, and then COVID happened, and the International Committee on the Taxonomy of Viruses was like, nope, we'll break and figure that out later. So maybe at some point they will make that a binomial classification schema. Okay, the differences again between eukaryotes and prokaryotes, we've already articulated the most important one, which is the presence or absence of a nucleus. Other important things though, to be able to contrast, eukaryotes have multiple chromosomes and they are linear. So if you were to stretch out all the DNA in the eukaryotic chromosome, it would not be a closed molecule, it would be linear. With prokaryotes, that is not the case. This cartoon kind of shows something, a compact molecule, but what it should be showing is that is a circular piece of DNA. That's what a chromosome is. A couple megabases in size, fairly large piece of DNA. In terms of replication, eukaryotes will divide by mitosis, which is where our chromosomes will divide, split apart, and go off into different daughter cells. And bacteria have a much simpler but similar way of dividing through binary fission. Basically, again, they're just making a copy of their chromosome, and that those separate copies of the chromosome are going off into the daughter cells. So we'll talk about that a little bit more as well. For energy production, eukaryotes will use our mitochondria to generate ATP in our cells. Bacteria will do this in their cell membranes. They have a lot of different cellular machinery to accommodate that. It's funny, I was grading an ACFT early this morning for the Department of Physical Therapy, quick tangent. And when they were planking, I was asking them, like, what their favorite bacteria is, just to take their mind off of it. And I kid you not, these young physical therapists both said mitochondria, favorite bacteria. Alright, so we have some work to do with those guys. But hopefully here in IPAPS we're coming in a little bit stronger on basic cell bio, and we know that mitochondria are not bacteria. We can get into the endosymbiotic theory, but that is not a bacterial cell. In terms of organelles, if you just look at the way these cartoons are depicted, it shows you that we do, like eukaryotes have developed and evolved a lot of complicated, specific cellular machinery. We have our mitochondria, we have our Golgi, we have our endoplasmic reticulum, lysosomes, vacuoles, a whole bunch of stuff going on. And again, most importantly, that enveloped nucleus that protects our genome. The bacteria, their stuff is all just kind of sort of sloshing around in the cytoplasm. They have a nucleoid, so it's kind of like a nucleus, and it's kind of organized a little bit, but not nearly as intricately as eukaryotic nucleuses are. So they have relatively few organelles, and overall just much more a simpler layout, simpler design. In terms of replication, briefly with prokaryotes, again this is just depicting what binary fission looks like. They don't have the very complicated checkpoints that eukaryotic cells have to go through when they're undergoing mitosis, but basically they get signals that it's okay to divide, and their machinery will start making copies of that chromosome, that single chromosome. And once you have a copied chromosome, the cell will start elongating. Very busy time for that cell. It starts making more proteins. It starts building up the cell walls so you can have enough material to make 2 full cells. And then in between, a septum starts forming, and those copies of his chromosomes will go off into each separate daughter cell. Oftentimes, when you're looking at bacteria under a microscope, you might see, like these are some bacilli right here in this image. And you can see where the septated partition is. Those still haven't fully detached from each other. They can stay attached like that for a pretty long period of time. So they may or may not eventually detach, but that's basically how they divide. Okay, I'm trying to think of how much I want to do today. You guys need a break? You doing okay? Great. Okay, alright, we'll drag on. It will be a short day today though. I think it's like, yeah, we'll keep today fairly short. Okay, so nomenclature, I know this is kind of a busy slide, but a couple things to point out here, especially when we get into talking about bacteria. We have some important taxonomic distinctions to make below the species level, starting with subspecies. We already talked about that with Homo sapiens, but with bacteria, you see subspecies much more commonly. An example of that, so used to denote distinct populations within a species, just a subspecies. But a really good example is Salmonella. So Salmonella is what taxonomic level is Salmonella, based on what we talked about earlier. Is it a
Genus.
It's a genus, correct, excellent. So we have our genus, and then we have Enterica, what would that be? Species, yep. Then we've got subspecies, abbreviated there, subspecies Enterica. This genus is a mess. There are like 2,600 different bacteria in that genus. It's a sprawling mess, and that's a reflection of the historical development of that genus. But basically, there's like a handful in one species called Salmonella bangori, and the rest of those 2,500, 2,600 are in Salmonella enterica. So at that point, it becomes useless to use that species level, and you have to bump down to the subspecies level, where you start getting into more interesting, informative stuff. So Salmonella enterica, subspecies enterica, and even that, you still have 6 major subspecies. So we have to bump down another level with this particular organism and go to a serovar level. Serovar is a contraction or a portmanteau of 2 words, serological variant. Means that on the surface of this bacteria, there are antigens that elicit different immune responses. That's what serological variants are. So Salmonella, this is ridiculous, but it's Salmonella enterica, subspecies enterica, serovar typhi. That's the full proper name. I didn't make it up. Fortunately, mercifully, you could shorten that to Salmonella typhi, where that serovar is not italicized and it's capitalized, and that's considered proper and short. But there's one example, and we will be talking about Salmonella. It's a major pathogen, so that's your intro to the horrors of the taxonomy of Salmonella. A little bit more accessible are strains. You've probably heard of strains with viruses and bacteria before, right? Strains are genetic variants or subtypes of subpopulations. So they have a common ancestor, but they are descendants that have traits that are meaningful and different and good to know. An example of that would be E. coli. So with E. coli, you have lots of different strains. This is a bacteria that you have in abundance in your gut, right? Most of that is completely harmless strains of E. coli. But there are strains like the K1 strain, which is associated with neonatal meningitis, or E. coli 0157H7. Anyone heard of that? Yeah? It's like a really nasty strain of E. coli. It's associated with hemolytic uremic syndrome in children. It can kill. It can be deadly. So lots of different strains. This strain I have up here, the K12 strain, is pretty much a completely avirulent, non-pathogenic strain. It's what we use in the lab, for those of you who've worked in labs before. That's a K12 strain. can do just about anything with it. Other non-pathogenic strains, DH5 alphas, XL10, like lots and lots of different strains. Okay, and then lastly, serogroups. These are groups of serological variants or serovars that have a common antigen. This is useful with a couple different examples here. You know, this slide is titled Prokaryotic Nomenclature, but we've got a virus down here because a lot of viruses are usefully thought of in terms of serogroups, like influenza A, H1N3, those are antigen groups that can be shared among different strains. But then the bacterial example up here is Streptococcus pyogenes. That is a species that has a common antigen on the surface of it, and that antigen is really useful for typing purposes in the lab. So clinicians, they might not know the scientific word, they haven't heard of Strep pyogenes, but they know what group A strep is. That's how common it is, and it's because it's useful in identifying those bacteria. More on that later, we'll hit that more. Also, antigens down here, if you haven't heard that term, it's something we'll be talking about a lot in this course. What are antigens? You want to put that in your own words.
Chemical marker on the surface of the cell that denotes what it is, and then it's an identifier.
Yeah, so he said a chemical identifier on the surface of the cell that can be used for identification and something else.
I just threw in for the immune system specifically.
Yeah.
In this context.
Yes, for the immune system. Did you have anything to add to that? No, okay. Yeah, so they can be useful for identification purposes. We have different antigens. This is, you know, a stylized bacterial cell, and spoiler alert, like everything on the outside of this pretty much is antigenic, meaning it can be recognized by the immune system and used to raise antibodies against it, for example. So we'll talk about that a lot in the next deck, but that's what antigens are. They're molecules that elicit an immune response. Okay, and then real quick, I will fly through this slide and put you guys on a wee break after that. This is one of those FYI slides. I don't have too many of these, but FYI means for your information, as in you do not need to spend a lot of time studying this or any time, really. It's not testable, but it just kind of explains some good to know things. Talking about Strep pyogenes, which we mentioned in the last slide as well. I said that was sometimes referred to solely by its antigenic group, group A strep. We'll hear that a lot. Sometimes beta hemolytic strep, group A beta hemolytic strep. Just know that there are a lot of different species of Streptococcus, and these different groups have a lot of different antigenic types in there. So we will talk about that much more in block 3. Salmonella, ditto, we've already talked about that. Shigella, ditto. These are all enteric pathogens, or Salmonella and Shigella are anyway. E. coli, O157H7, mentioned that as a strain, very, very common. But the thing I really want to draw your attention to in this slide is this bullet about the use of colloquial names for bacteria. You will hear this a lot when you go out and practice medicine, but no one ever actually says like Staphylococcus aureus, right? They say staph or Staph aureus. So there's your colloquialisms that I use when I lecture, and you can kind of get used to start hearing some of these if you're not familiar with them already. Another very common one is C. diff. Anyone know what C. diff is? Yeah. A lot of you guys. What is it? Probably. Yeah, it does have like barnyard smell. What is it? Clostridium and the species difficile. Yeah. So nobody ever says like Clostridium difficile. It's a mouthful, and it's actually been recategorized into a different genus. Now it's Clostridioides difficile. People just call it C. diff. So you hear that a lot. Tuli. Anyone know what tuli is? What bacteria? Yeah. So it causes tularemia, and the bacteria is Francisella tularensis. Oftentimes it's called tuli. Trick. Anyone? Or tricky? Huh? No, but put a pin on that. Trichurius, that's a worm. Yeah, so whipworm. And then this one, so T-vag, that's the one you were saying, I think, yeah, so Trichomonas vaginalis, like, people just like calling it T-vag instead of Trichomonas vaginalis, but it's an STI, what else about it? Yeah, very common STI, causes trichomoniasis. And the last thing on this slide, covered non-italicized, we're gonna say species, just abbreviation. That's it. Let's take a well-earned break. I've got 13:50, start back up at 1400.
It's a yeah, it's a what's the bacteria? What it does. It's still considered like it's a trick because the bacteria. It's a hard bacteria, which is that's in itself. Like the infection as a bacteria.
Yeah.
Sure.
Yeah, that's fair. So we know we were talking about that. There's been times when like it's live on their like species. So yeah, we don't know if it's live or not. It's gonna take effect. So you can turn off the plug next year. Like just on the I don't want to say consumer end, on the front end, say under flash cards, this material and like close solution. Here are some examples I want you to follow. You do it on the front end as opposed to like through the back.
Some of those you post to ship on, we got a total 4,000 passage. Oh my God. It was very frustrating. It was really nice.
So trying to fish for that right answer, which yeah. Like, yes, it is, but whatever's easiest for you. I think that you can kind of play around with.
Yeah, I can put it in there. I don't know what else to put it in besides like the class.
Yeah.
I'll put it on underneath of it.
Yeah, I appreciate it.
Yeah. I didn't make sense. Did you really? Alright, so yeah. I was like, for a test, I
This gentleman right here. He's the man. Man, 6.
Last, every single semester, last semester. Semester before was there. You guys coordinate.
I was gonna put it farther back so you can see the instructors, but the video's not great, like everything is blurry.
Oh, yeah.
But it picks up good audio
Yeah.
there.
Yeah. Don't show the instructions.
Right, like this.
Nice long 10-minute break. 9.5 minutes. Alright, let's just do a few more slides and then we will get you out here fairly early. That sounds good. Same with tomorrow. Okay, well, a couple slides talking about characteristics of scenarios. I've got my little laser pointer here. Yes, you can see. Okay, cool. so we've gone over most of this on that one comparison slide, if you remember a couple slides back, prokaryotes, eukaryotes. But we've got, now that we're looking at a giant cartoon or figure, bacterial cell. As I said, first thing we want to note is in the name, prokaryotic, no nucleus, got all of its genetic material, all of its DNA kind of compacted, supercoiled, but not in a membrane-bound nucleus. So out in the center of the cytoplasm here, not a lot of organelles like we talked about, but there are ribosomes. We haven't talked about that yet. You guys know what ribosomes do? Protein synthesis, yeah. So they have them too. They've got to make proteins. They are different than ours, but they do the same thing. Different enough from ours where they are a good antibiotic target. So we'll talk about that more later as well. The cell wall is probably the most important feature. So we'll spend a good chunk of time talking about the cell wall, not just in this block, but also in block 2. And that's useful for a number of different reasons. It's where a lot of antigens are for typing and identification. It's a good antibiotic target. It's how we group these, categorize them, so it's worth studying that. We talked about the diverse metabolism, but this is not a academic microbiology course, so we're not going to sit here and talk about how they metabolize different nitrogen sources or sulfur sources or carbon sources. Just know that bacteria have diverse modes of metabolizing substrates. And then rapid reproduction. Bacteria are simple life forms. Doesn't take much for them to divide. Some of them take longer than others, but on average, a cell like, or species like E. coli takes about 20 or 30 minutes to divide under ideal optimum growth conditions. So you can do the math, or we'll do it together in a couple slides, but that builds up very quickly, exponential numbers of bacteria. In terms of the morphology of bacteria, what do we mean by the word morphology? The appearance, how it looks to us. We use microscopy to look at the appearance, and this would be an example of a gram-stained specimen. And then we can use macroscopic morphologies to help us with identification as well. With both of these specimens, or both of these types of morphologies, we can see that we have very mixed populations of bacteria, so there's further work to be done. But on the left here, basically, we're seeing 2 different types of stains that we use in microbiology very commonly: gram-positive bacteria and gram-negative bacteria. So this circled bacteria down here that has kind of like purplish color, what is that? Is that gram-positive or gram-negative? Gram-positive, yes. And then up here, it's very hard to see, but what do you think? What color is it, first of all? It's like reddish pink, yeah. So is it gram-positive or gram-negative? Gram-negative, yes. So if we have like a dirty specimen type, like fecal matter or urine, not clean catch urine, wounds, things like that, they oftentimes look like a very messy specimen when you look at them under a microscope. So gram stain is one way to get a quick glimpse of information about what kind of bacteria are in those specimens. And here on the plate, similarly, who knows what the specimen source is, but whatever it is, it grew up a lot of different types of bacteria, a lot of different morphologies that we call morphotypes. So each one of these different morphologies is numbered, and we have a whole set of pretty developed vocabulary to describe these characteristics. You know, what color it is, what size, what shape, how shiny it is, if it's translucent, raised, concave, bubbled up, all these things that we describe bacterial colonies with. Going back to how they look under a microscope, they have different shapes. So this slide summarizes and lists those main different shapes. Coccus is what we'll be studying first when we start block 3, specifically Staphylococcus and Streptococcus. The coccus simply means spherical. So for the most part, these are fairly round. They like to be a little bit squished. The plural is cocci, and these bacterial spheres or cocci can be They can be in chains, which we don't really see in that image here so much. Clusters, tetrads, groups of 4, all sorts of things. Bacillus is the other major shape for bacteria. It means rod-shaped or cylindrical. That would be something more akin to this bacteria we can see down here. Rod-shaped, and from there it gets a little bit messy, but if you know nothing else, know that cocci are spheres and bacilli are rods. Cocobacillus, that means that it's somewhere in between, just short, stubby rods or squished, elongated cocci. Vibrios, that's a genus of bacteria, but that's also a shape. That's talking about bacteria that have this kind of curved shape to them. I don't have a picture on the slide that shows the Vibrio shape, but it's curved or comma-shaped. Carineiform, or diphtheroid, means that it is a rod, but it is asymmetric and kind of loaded on one termini of that rod. So they end up looking a little bit club-shaped. And they also have a unique morphology when they're clustered together. So they're often described as palisades, where they're stacked next to each other like that. That's called palisading. Spirochetes, very distinctive. That's what we're looking at down here. You see that super long bacterial cell there. They're many, many, many times longer than they are wide. And they have a built-in flagellum that gives them that sort of wavy appearance like that. So we'll talk about a couple clinically relevant spirochetes, syphilis, Lyme disease, and a couple others as well. Spirillum, that's another shape. We won't be talking about any spirilla in this course, but they also have kind of a funky spiral shape, except they're tighter than that. They're almost corkscrew spirilla. And then lastly, you gotta know the term pleomorphic. It's kind of a frustrating word, but it basically just means can appear in different shapes. Same species, depending on how old it is, what kind of shape it's in, if it's unhealthy, healthy, pleomorphic. So oftentimes due to environmental conditions that influence the shape. And here you can kind of see those cartooned out cocci that are in different arrangements, bacilli that are growing up in chains, cocci in chains, and those palisading diphtheroid carineiform bacteria that I mentioned. In terms of size, there should be a link, I hope, in your deck for some website that gives you a scale of bacteria. It's not testable, but it is a fun tool. It allows you to kind of zoom out from a macroscopic object like a pin, I forget what it is. Sorry, zoom in from there all the way to the size of like tiny ribosomes and organelle-sized things. So somewhere in between in that scale, you will hit the size range of bacteria. Most of the bacteria we're talking about somewhere in the range of 0.4 to 0.8 micrometers. That funky looking U, that's a mu, the Greek mu. So the millionth of a meter. Way, way, way too small to see with the naked eye. The bigger ones might get up to 1.5 micrometers in diameter, like Staph aureus, for example. Coccus, so you know it's spherical, so that's a pretty big sphere, pretty big cocci, 1.5 micrometers. So those are very satisfying to look at under a microscope because they're so big and easy to identify. You look at something like Pasteurella, and that is much harder, even with a relatively high-powered objective. Pretty tiny, 0.2 to 0.7 micrometers. So that's a bacillus and just much, much, much smaller. But that gives you a sense of the approximate size range of some of these bacteria. All right, in terms of structure, we've got the cytoplasm. Not that you would ever need to know what the texture is like, but it is sticky and sappy because it's got a lot of carbohydrates and other it has sugars, it has lipids, it has proteins in it, a lot of stuff in there, so it's very viscous. And in it, it has the genome that we talked about, the DNA, a chromosome. It'll have ribosomes. It will have a lot of enzymes and proteins that are responsible for synthetic pathways, making macromolecules, just the normal business of being a cell. All of that is in the cytoplasm. Moving outwards from the cytoplasm, we've got the cytoplasmic membrane. So this is very much like our own membrane, our own phospholipid bilayer, and our eukaryotic cells. If you look at the cartoon here, we've got, you can see phospholipid bilayer, so those hydrophobic tails of those lipids are oriented inwards, where they're touching each other, and then the hydrophilic components, which you may have already talked about in biochemistry, I don't know where you are in that, but those are oriented outwards. And the intent there is to kind of create a little bit of a barrier between the cytoplasm and the exterior of the bacterium, but there is a wall that is on the outside of that cytoplasmic membrane as well. In that membrane, we've got a couple important things that are going on. We've got porins, these proteins that function as channels in that cytoplasm, or cytoplasmic membrane, and that allows nutrients to pass into the cell and waste products to pass out of the cell. You've got carrier proteins and you've got enzymes. Where do we say that energy is produced in bacteria? Cytoplasmic membrane, yeah. So like those integral proteins, a lot of those would be involved in energy production as well, electron transport chain. So these are all the functions. We've talked about all of these, I think. Site of energy production. Bear in mind this is for eukaryotes, ATP produced in mitochondria eukaryotes, but cytoplasmic membrane for prokaryotes. Very importantly, synthesis and transport of peptidoglycan. Talk about that in the next slide, but that occurs, it's like prefabricated here in the cytoplasmic membrane, sometimes called the peptidoglycan factory, where all of that is pre-assembled before it's moved out of the cell wall to be added into that structure. You've got teichoic acids, and other proteins that are in that outer membrane. Okay, this is a busy slide, but it's super important, and yes, sir.
So, if you go back to the last slide, so energy production in the cytoplasmic membrane, and if you go back to the slide before that, it says energy production mechanisms in the cytoplasm. Is there a differentiator there, or is it just the bulk of the energy is produced in the?
Yeah, I think energy production mechanisms, that is super confusing. It should, like the bulk of your ATP production is going to be in the cytoplasmic membrane. Other biosynthetic pathways, yes, in the cytoplasm, but yeah, I think strictly speaking, this is probably confusing and not quite right. So thank you for pointing that out. Yeah. Okay, so the cell wall. The cell wall is on the exterior of the cytoplasmic membrane. The function of this is to provide shape and rigidity to the bacterial cell. Make sure it can be protected from the environment, and it also helps with attachment to whatever substrate these bacteria are trying to attach to. So there are a lot of antigens on the cell wall, so those are important too, and we'll talk more about those later. But when we're talking about gram-positives versus gram-negatives, these are some of the key differences, the key structural differences in the cell wall that you need to be familiar with. With gram-positives, they've got their cytoplasmic membrane, so if you're staying with me, that's this layer that's on the next to the interior of the cell, right there. And then above that, you have space, periplasmic space. And above that, you have the peptidoglycan layer. So peptidoglycan is just a bunch of carbohydrates that are strung together, and it provides structural support for the cell. You also have teichoic acids that are present in gram-positive bacteria, but not in gram-negative bacteria. Those teichoic acids will help maintain structural integrity with that layer of peptidoglycan. They assist with that, as do lipoteichoic acids, which go a little bit deeper and they extend past that periplasmic space into the outer membrane. With the gram-negatives, very different structure, a little bit more complex there. But you have that cytoplasmic membrane, then you have a periplasmic space, and then you have a layer of peptidoglycan, but that layer of peptidoglycan is much, much, much smaller or shorter than it is with gram-positives. So So with gram-positives, you might have a layer that's 50 or 60 different monomers deep with that cell. With gram-negatives, you might have just a few deep, so much smaller layer of peptidoglycan, then another paraplastic space, and then we have the outer membrane. So with that outer membrane, we have some other important characteristics. Lipopolysaccharides, or LPS, this is commonly abbreviated, and the LPS will have lipid A, which is really, really important in terms of pathogenicity because it can be toxic to us when we encounter large amounts of lipid A. And then O antigen, which is an antigen that is very, very useful for identification purposes. We use it a lot for epidemiological purposes. And then porins as well. With both of these different cell walls, gram-positive and gram-negative, you can have bacteria that express another layer called capsule. We'll talk more about that later as well. But that's what's kind of shown by this blue-looking blob on both of these figures. That's capsule production. Not all bacterial species produce capsule, but those that do will have this thick mucoid-like layer protecting them as well. With peptidoglycan, we mentioned that on the other slide. Which type of bacteria had a thicker layer of peptidoglycan? Is it gram-positive? Gram-positive, okay. So peptidoglycan provides a lot of that structural rigidity and strength to those bacterial cells. Structurally, you have these long alternating chains of N-acetylglutamic or glucosamine and N-acetylmuramic acid, NAG and NAM. We just call them NAG and NAM for short. So these figures kind of show you or give you a sense of how those monomers or those chains of NAGs and NAMs are strung up together in 3 dimensions to provide that rigid support, but still give you enough flexibility where that bacteria can squeeze through things, it can attach with some flexibility. So there's a little bit of give and movement. It's kind of analogous to having like a chain link fence, which you can come up and shake really vigorously, but then stack that 50 or 60 layers deep and connect those together. So that's kind of like the structure or the idea of peptidoglycan. It's all reinforced together in a mesh-like structure. We do have some antigens that are useful for bacterial identification, particularly with gram-positives. And most importantly, for you guys in particular, you gotta know that peptidoglycan is a major target for several classes of antibiotics. Okay, so we've covered cytoplasm, the membrane, cell wall, a couple other structures to cover with bacteria. First, we'll talk about that capsule that I mentioned when we were looking at gram-positives versus gram-negatives. Both of those types can produce it. And basically, it's just like a really thick gelatinous layer of polysaccharides that is produced and moved to the outside of the cell for the purposes basically of evading immune detection a lot of times, surviving in a host. So a lot of times bacteria that produce capsule are pathogenic to people because they have all these features from capsule production. It makes them resistant to phagocytosis, which we'll be talking about next in the next PowerPoint deck a little bit more, but phagocytosis is the eating of those cells, those bacterial cells, immune cells. Capsule also masks some of those surface antigens that would otherwise be eliciting an immune response, and it assists with adherence to host cells and tissues. On this plate, you can see how slimy and almost like glistening a mucoid colony is. So you can actually see that macroscopically when you grow up a strain that produces capsule. It looks like a shiny, kind of snotty colony. And you can even see that on a gram stain. This white area around those bacterial cells, that's capsule. So that gram stain isn't penetrating around that capsule. It's basically staining white. That's all capsule protecting those bacteria. And non-mucoid colonies, you can see like down here, some non-mucoid colonies. Those aren't shiny at all. They don't express that capsule. I mentioned that in gram-negative cells, you have the LPS, the lipopolysaccharides, right? If you look at the structure of lipopolysaccharides, you have our O antigen on the outside, just a bunch of polysaccharides. Core polysaccharide, which is useful for structural purposes but not contributing to pathogenicity, not useful for typing, so we won't focus on that too much. But then at the very bottom end, when you're getting into the outer membrane of that gram-negative, you have the important part, and that's lipid A. That lipid A has a bunch of fatty chains of lipids extending off of these carbohydrates. And what that does clinically is when you have the rupture of bacteria in a person who's sick with bacteria en masse, like you've given them a heavy dose of antibiotics and you have massive die-off of gram-negative bacteria, all of this junk gets released into the body before it can be handled properly by the immune system, and that results in some heavy immunopathology, things like fever, septic shock, because you haven't had time to deal with lipid A. So that's an important part. Again, in gram-negative bacteria only, super important. We'll talk about that again, but make sure you are aware of what endotoxin is.
Is the picture of the LPS oriented correct? Like with what it would be on like the surface?
Yeah.
So the
Yeah.
lipid A is sitting more internal.
Exactly.
Okay.
Yeah, so we've got our gram-negative bacteria. We're zooming in on the cell wall of the gram-negative, and then the LPS is on that outer membrane. So the lipid A part is the proximal part of that LPS, and that O antigen would be the distal part, if you will, kind of on the outside or the environmental side of that bacterial cell. All right, flagella. Probably all seen pictures of flagella before, but what do bacteria use flagella for? Motility, yeah, primarily motility. But as we've talked about before, if it's on the outside of a cell, it is antigenic, right? It's something your immune system can see. So flagella are important for antigenic reasons, developing antibodies against them. And in terms of morphology, different bacterial species have different arrangements of flagella. You can have a single flagella coming off of one termini of a rod. You can have tons and tons of flagella coming out from all directions. Paratrichus, hypertrichus, there are lots of different arrangements of flagella, but basically all you need to know is that they provide motility for that bacterial cell, and they can be controlled by the bacteria and moved around to propel the bacteria in some direction. So the bacteria, for example, they can sense nutrients and kind of steer themselves in a gradient, it's called chemotaxis, towards those nutrients. Or if there's a threat, they can steer themselves away using their flagella. These are only for rods, for your bacilli, so unfortunately cocci do not have flagella. They can't move themselves away. But pretty straightforward. There's a scanning electron micrograph showing a bacterial cell, tons of flagella coming out from all over, all directions. More appendages, a little bit different, but similar. Fimbriae and pillae. Fimbriae are usually described as short hair-like projections on the outside of these bacteria. They have a different role, not so much useful for motility, but they are involved in touching those target cells, those host cells, and forming attachments. So important in adherence and colonizing a host. Pillae are also appendages, but you don't have as many, and they're involved in a very specialized process, bacterial conjugation. So that's what you're seeing in this image right here is bacterial conjugation facilitated through a pilus, which is singular pillae. This is called a sex pilus. Not quite analogous to what you would think of as sex, but there is a donor and there is a recipient, and the donor is throwing that pilus out towards the recipient with the overall objective of sharing and transmitting genetic information, right? So that's what's happening here, and that is facilitated by the pillae. Some more pretty pictures of fimbriated bacteria, very long, loopy structures there. And then, just see something really quick. Yeah, we'll just do this last slide and we'll call it a day. Okay, so endotoxin. endospores. Not all bacteria produce endospores. In fact, there are only two genera, which is the plural of genus, genera. Two genera of clinically significant bacteria that do produce these endospores. One is Bacillus, which has members like Bacillus anthracis, you've probably heard of that before. Causative agent of anthrax. And the other is Clostridium, which we've already talked about, Clostridioides. So those two genera can form endospores. The purpose of these things is basically for survival, right? It allows these bacteria to withstand some incredibly stressful, adverse environmental conditions. So those bacteria will be out doing their thing in their metabolically active form, which is called, ironically, the vegetative state. So they're dividing, they're taking in nutrients, etc. And they get environmental signals that times are getting rough. So there's heat, there's starvation, there's desiccation, there's dryness, something like that. And they get a signal to start sporulating. And they turn on a bunch of genes that basically shut off metabolic activity and start forming this really dense husk around the genome. So that is the purpose at that point. You want to save the genome. That's the whole purpose. So you form that husk, the rest of that vegetative cell basically just dies off. It goes away, and all you're left with is this metabolically inert spore at the end of the day. These spores can persist out in the environment for days, weeks, months, sometimes years, long time. So the implications for that are when you have spore-forming bacteria, it is hard to disinfect them, it's hard to kill them, it's hard to penetrate. If you're using some sort of disinfectant, it's very hard to penetrate that husk or that outer wall with disinfectants. Treating them with antibiotics, like if you have a patient with Bacillus anthracis, who has anthrax, and so those bacteria have sporulated, those antibiotics have a very hard time penetrating through that spore. So those are some of the implications of spore-forming bacteria. But fortunately, like I said, only two genera can do it, Clostridioides and Bacillus. But still, important to know. Okay, I think we'll stop there for the day. And then we'll pick up on this slide tomorrow. Any questions for me? Yes, sir?
Just to confirm, Matt, you said that for the endospores, the vegetative state is when it's like metabolically, biologically active.
Yep.
And then the spore state is when it's shutting itself down.
Yes, exactly. So that Bacillus that you see there, that would be the vegetative cell. It's already turned on genes to start sporulating. When and if conditions become favorable again, it will do the reverse process, which is called germination. Then it germinates back to a vegetative cell that can actually resume those metabolic activities again. Yep. Any other questions? Okey-doke. All right, guys. Well, I'll let you go. Have a wonderful rest of your afternoon. It's a pleasure to meet you. Looking forward to getting to know you better. And I'll see you guys tomorrow at